Welcome back to another video PharmCon tutorial. In today's lecture, we will discuss about penicillins. Antibiotics are agents that kill or inhibit the growth of other microorganisms. In this lecture, we will specifically talk about beta-lactam antibiotics and particularly penicillin in detail. First, let us see why they are called as beta-lactam antibiotics. They basically have a beta-lactam ring. Lactam is basically a cyclic amide. This beta-lactam has a carbonyl group and an amide group. This is called beta-lactam because the nitrogen group is attached to the beta carbon relative to the carbonyl group. It also has a sulfur-containing thiazolidine ring. Bonds in this structure is very dense and tends to break very easily. The bond most prone to break is one between the amide and the carbonyl group. Now, the beta-lactam antibiotics include penicillin, cephalosporins, carbapenems and monobactam. In this video, we will discuss only about penicillin. First, let us have a look at the structure of penicillin. As mentioned earlier, they have a beta-lactam ring and a thiazolidine ring having a sulfur group which is responsible for the hypersensitivity reactions associated with this penicillin. It also have a R group, and this R group can be modified and the natural penicillin can be converted into semi-synthetic forms, as well as they can be modified such that they become protective from beta-lactamases and gastric acid. But while doing so, there are some consequences too. That is, it will lead to increase in the molecular weight of penicillin, and thus there will be decreased penetration of penicillin through the porins of the bacterial cells, and thus a very narrow spectrum antibiotics will be produced. Now, we know that the gram-positive bacteria have very thick peptidoglycan cell wall, and the porins are attached to the membrane lipids, unlike the gram-negative bacteria which are present in the outer lipopolysaccharide layer. This gram-positive have an inner plasma membrane, and in between these two layers they have the periplasmic space. In contrast, this gram-negative bacteria have an outer lipopolysaccharide with porins, and there is a very thin layer of peptidoglycan. Between these two layers is the periplasmic space, and then there is the plasma membrane. As we know that these penicillins and cephalosporins are bacterial cell wall synthesis inhibitors, so they are more effective on gram-positive bacteria because they have a significant well-established peptidoglycan layer than the gram-negative ones. Now, why is the bacterial cell wall so important? What this cell wall does is that it gives support to the bacterial cell, it protects the cell, it maintains the shape, and it also allows the bacterial cell to withstand low osmotic pressure and prevent from cell lysis. Now, moving on to the mechanism of action of penicillin. These are bactericidal agents that leads to bacterial cell lysis by inhibiting actually the synthesis of cell wall. Now, we know that the bacterial cell wall is made up of peptidoglycan, which has a glycan chain cross-linked by a peptide chain. This glycan chain is composed of alternating amino sugars like N-acetyl muramic acid and N-acetyl glucosamine. To this N-acetyl muramic acid, a pentapeptide is attached which has terminal 4 and 5 D-alanine amino acids. To its third amino acid is attached a pentaglycine chain, and this pentaglycine chain is needed to bond to the 4 D-alanine amino acid of the another N-acetyl muramic acid. While this bonding has to occur in the presence of enzyme transpeptidase. This process also cleaves off the terminal 5-D-alanine amino acid. Now, this penicillin have a structural analog of D-alanine, and this is why they are able to inhibit the transpeptidase enzyme, inhibits this bonding and thus diminish the cell wall synthesis. Now, one thing to remember is that the penicillins and other cell wall synthesis inhibitors only work in dividing bacteria. This is because these cells will be synthesizing cell walls and provide a target for the penicillins. By inhibiting the cell wall synthesis, they actually cause the bacterial cells to lyse and ultimately have a bactericidal action. There are many bacteria that are resistant to penicillin, and this is attained by three main mechanisms. First is via the production of beta-lactamases, and these are the enzymes that cleaves the specific susceptible bond in the beta-lactam ring of the antibiotic. These type of resistance are common in the Staphylococci, E. coli, etc. Next mechanism of resistance is by altering their penicillin binding proteins or the transpeptidases, and thus they can evade the penicillin and not let the penicillin to attach to the penicillin binding protein. And this is mainly seen in methicillin resistance. The third mechanism of resistance is by change in the porin structure. The bacteria will change the size or shape of their porins, and so the penicillins cannot enter the bacterial cells, and these type of resistance are mainly seen in the Pseudomonas. Let's have a look at the classification of penicillins. This classification of penicillins are made on the basis of the spectrum of activity and also sensitivity to the beta-lactamases. First one is the narrow spectrum antibiotics, and they are actually sensitive to beta-lactamases. Second is the very narrow spectrum penicillins. They are actually beta-lactamase resistant. Next is the broad spectrum penicillins. They are beta-lactamase sensitive. And finally, we have the extended spectrum, which are also sensitive to beta-lactamases. The first two, that is the narrow and the very narrow agents, are not given without any protection to the beta-lactam ring, and the last two are given with protection. Now, what do we mean by the protection? By protection, we mean that we administer beta-lactamases inhibitor along with the broad spectrum and the extended spectrum antibiotics to actually save them from the beta-lactamases. Now, how do they work? They actually resemble structurally the beta-lactam molecules, and they bind to the beta-lactamases and then be converted into the metabolites, and these metabolites inhibits the beta-lactamases irreversibly. These beta-lactamases inhibitors include clavulanic acid, sulbactam and terabactam, and these are available in combinations with antibiotics. The examples for these combinations are augmentin, timentin, sulbacin and tazobit. Now, looking back at the classification of penicillins, the first narrow spectrum antibiotic is the penicillin G. It is well distributed in the body but has poor penetration to the blood-brain barrier. The duration of action is about 4 to 6 hours. Next is the procaine penicillin G with 12 to 24 hours action, and we have benzathine penicillin G and penicillin V. Now, among these, the first three are not effective orally because they are acid labile, that is, they are destroyed by the gastric acid, so they can be given by IV or IM route. This penicillin V is acid resistant and can be given orally. Now, about its uses, the bacteria that is still susceptible is the T. pallidum, which causes the syphilis, and they can be also used against staphylococci, pneumococci and even meningococci. The second class is the very narrow spectrum, and they include naphicillin, oxacillin, cloxacillin and methicillin. These are given when there are known or suspected infections by staphylococci that is not methicillin resistant. They do pay a cost of beta-lactamase resistance, To make these agents beta-lactamase resistant, they are substituted with bulky R groups and thus makes it susceptible by saving the bond from breaking. But actually by doing this, the molecules becomes very large and thus there is less penetration through the porins, which makes them very narrow spectral antibiotic. Next is the broad spectrum beta lactamase sensitive agents, and they include aminopenicillin, that is the amoxicillin and ampicillin. Now both of them have some differences. Amoxicillin have a very good absorption from the gut, while the ampicillin is not that readily absorbed and have high concentration in the gut, so they are used in the case of dysentery associated with the Shigella. This amoxicillin causes decreased side effects when compared to the ampicillin because it is having very low concentration in the gut, so it won't harm the natural flora and has a decreased incidence of diarrhea. This amoxicillin has no food interactions, but in ampicillin they do interact with the food. Now about the uses. Amoxicillin being a broad spectrum antibiotics, they are used in both penicillin sensitive and resistant streptococcal pneumonia and also against gram negative bacteria. And about the ampicillin, they are used in listeria and the shigellosis. Last about the penicillin is the extended spectrum, which includes the carboxypenicillins and the uridopenicillins. These agents are also called as anti-pseudomonal penicillins because these organisms are very susceptible to these antibiotics, and which includes the ticarcillin, then piperacillin, then carpenicillin and maslocillin. These extended spectrums are mainly used in the used against this Pseudomonas aeruginosa, then protease infections, and even in the treatment of UTIs, burns and hospital acquired infections. Finally, about the side effects of penicillin, we know that the thiazolidine ring of penicillin has a sulfur group in it. So anywhere if there is a sulfur, there is hypersensitivity reaction. So before injecting this penicillin for therapy, we first need to do a sensitivity test by injecting penicillin intradermally, and if there is a reaction, we should stop and not proceed to penicillin therapy. And mainly this hypersensitivity reaction is seen in natural penicillin, that is the penicillin G. Another problem is the cross allergenicity. That means if a person is allergic to one form of penicillin, then there is a high chance of allergenicity to other forms of penicillin. Another side effects is the GI distress, and this is mainly due to the destruction of the normal flora of the gut. Lastly, this Jerish Herxheimer is yet another complication associated with this penicillin therapy in syphilis treatment cases. When penicillin therapy is started in a syphilis patient, the bacteria starts to die and it releases the enterotoxins, which causes acute exacerbation of the signs and symptoms of syphilis like fever, chills, myalgia, etc. And in the past, this was used as the marker of effective treatment, but now it is just a side effect, and which is treated with aspirin and corticosteroids.